Last part of protein synthesis, last little part of your DNA. So this is now the second phase of protein synthesis. So last time we kind of met, we went through the process of transcription. And what transcription is we are now taking DNA's code from the nucleus, transcribing it into a smaller message and sending it out into the cytoplasm, which is where proteins are going to be made. So this phase of protein synthesis is now called translation. We need to translate the language that messenger RNA made from DNA into your protein. Because remember, DNA code was too big. So messenger RNA went in, put it into its own language, brought it out to the cytoplasm, and now transfer RNA is going to help to take that language that messenger RNA made it into and change it into the code for the protein. So all your amino acids are going to be strung together. So this phase of protein synthesis is now going to take place in the cytoplasm. The first phase, transcription, since it involved DNA, is going to take place in the nucleus. So there's kind of a lot going on because we said we have messenger RNA involved here. It got made within the nucleus, single-stranded. It came out through the nuclear pores because it's much smaller, and there it hooked up to a ribosome. The ribosome, we said, had to be activated. You had a large subunit, you had a small subunit. Both of them make up the ribosome. They have to kind of come together, and that's what our RNA has to do. So our RNA is kind of like the glue that puts them together so that it can read the messenger RNA strand, and it's also going to activate it or turn it on so that it can read the codons within messenger RNA. You also have transfer RNA. So your transfer RNA was also made in the nucleus. It traveled out into the cytoplasm. We said those are the ones that look like little T's or little cloverleaf structures, and they're floating around that in the cytoplasm, and they pick up your amino acids. So we know that they can pick up certain amino acids. They can only pick up specific amino acids, and that's going to be based upon the three base pair code on the bottom of them, and that is their anticodon. So I'm going to kind of backtrack just a little bit there. There's my transfer RNA that we just talked about. Carries amino acid. It knows which specific one to carry based on its three base pair code, which is called an anticodon. Remember, it doesn't matter if it's messenger RNA or transfer RNA. Any type of RNA, you are now going to have uracil instead of thymine. There's no thymine that we're going to have within this process of protein synthesis. So as we go through, now we've got these transfer RNAs and they're out in the cytoplasm. They have to kind of go through and pick up and become activated, pick up their amino acid and become activated. And this is going to take energy. So we've talked about this ATP molecule. This is the energy that's going to be required. So ATP is kind of split in half. Well, it's not really split. Those phosphates, when we break those phosphates off, that energy from those phosphates are going to be activating and attaching that amino acid to your transfer RNA. So once again, I'm going to go back. This picture here, in order to get this amino acid attached to my transfer RNA, it takes ATP. So ATP comes in, attaches that energy. There's now a bond here between the two of them, and that ATP molecule now becomes AMP, adenosine monophosphate. So we broke off two phosphates and stored that energy within the bond. This happens at a giant enzyme. And this enzyme complex that this transfer RNA and amino acid become attached to by energy is called amino acyl synthetase. It's actually amino acyl tRNA synthetase. So when I do the poster project, you guys are going to see these giant enzymes that are going to go through and be seen, and that's where transfer RNA and your amino acid are going to become activated and attached with the help of that energy. So at that time, when this happens and they're within that particular enzyme, that active site of the enzyme, the amino acid is now activated. And transfer RNA is going to transport him to the correct location on that messenger RNA strand. So when that amino acid AMP, because now we've switched that high energy molecule to a low energy molecule, enzyme complex meets the tRNA molecule, we basically are going to go through and have this activated molecule now. So we talk about these 3' and 5' ends because remember, it's not only your messenger RNA strand that has 3 and 5' ends, it's not only your DNA strand. Anything that has sugar in it is going to have this. So we know that the bond is going to form between the amino acid and the 3' end of transfer RNA. So let's take a quick look. This end of my transfer RNA is going to be my 3' end. Sugar, phosphates, nitrogen bases. It's still sugar and phosphates all the way down. Here's the nitrogen bases. my 3' carbon is facing up, and that's where my amino acid is going to attach to with the help of the ATP energy and the enzyme amino acyl synthetase. So when transfer RNA is now it's going to become bonded to messenger RNA, the codon and anticodon are going to be attached just briefly, and the amino acid is going to be brought in the correct place. We're kind of jumping ahead just a little bit there. As they do this, my amino acids are going to start to string together, and they're going to be joined together by what is known as a peptide bond. We talked about this before. We talked about it in organic when we did proteins, and we said, okay, well, proteins are made up of specific amino acids. Amino acids can go through and they can bond together with a peptide bond. 2 amino acids joined together is a dipeptide. A whole bunch of them is a polypeptide, and a polypeptide is just another name for a protein. So when we go with this process of translation, so at this point, messenger RNA has left the nucleus, it's out in the cytoplasm, and my whole process of translation is going to start. We're going to break it down in 3 steps here. So we have initiation, elongation, and termination. So when we look at initiation, this basically means start. The small ribosomal subunit, the bottom portion, attaches to the messenger RNA strand near the 5' end. So far we said, okay, messenger RNA is going through, and messenger RNA got made 5' to 3'. The old DNA got read 3 to 5. Same way replication occurs. Now messenger RNA is going to enter into the ribosome. Now messenger RNA is going to get read 5 to 3. So that's opposite of everything that we've said so far. So my 5' end of my messenger RNA strand, that's where it started getting made, because remember, it got made 5 to 3. So this 5' end is now going to enter into the ribosome. The first codon on that messenger RNA strand is called the initiator codon. So there could be 100 base pairs that go through this ribosome, because remember, he's activated and he is reading my codons. But until that ribosome reads AUG, nothing's going to happen. So AUG is the initiation or start codon. So it doesn't matter what goes through that ribosome, U, C, G, C, G, until it reads the 3 base pairs AUG, nothing happens. As soon as it reads AUG, it starts calling out, oh, AUG, AUG, I need to find my anticodon that's going to be complementary to that. So AUG is my 3 base pair, my start codon in my messenger RNA strand. At that point, the large ribosomal subunit is going to lock into place with the help of rRNA, and now tRNA is now going to start coming in. So within your ribosome, there are 2 subunits on this, and the 2 subunits that we're going to see. So the 2 subunits that we are going to see of my ribosome. So I went and kind of grabbed this. This is an example of what a ribosome is. Here's my small subunit. Here's my large subunit. So what's going to happen with this ribosome that is out in the cytoplasm? Messenger RNA that got created is now going to come through, and it's going to enter into the ribosome. So just to give you guys a quick little view, messenger RNA enters in. This is actually my 5' end. If we had blown it up and we looked at the sugar, my 5th carbon would be facing towards the end here. So my 5' end is going to enter in, and until, like I said, there could be thousands of base pairs before then that entered into it, but until it reads AUG, nothing happens. So that ribosome, my small subunit, is going to read, oh, it's got my codon AUG. Large subunit, small subunit come together, they become activated, and they are ready to read my messenger RNA codon now. So we're going to stay in the first location on my ribosome. So what you're going to see here is that there are 2 spots on my ribosome. As that messenger RNA enters in, it enters the first spot on the ribosome, which is the A spot, or the A site. This A site is known as my activation site. That's basically where I read the start codon and everything starts to occur. The second site is my peptide site. So what's going to happen? Out in the cytoplasm, we just said I have an enzyme called amino acyl synthetase. That amino acyl synthetase is going to help to bond or activate my transfer RNA as well as my amino acid. So here's what my transfer RNA looks like. It's kind of clover leaf shape. On the bottom, you guys are going to see 3 base pairs. These 3 base pairs make up my anticodon. At the top end, which is my 3' end, I am going to attach to it, or it's going to pick up a specific amino acid, and it knows which ones to pick up based on the 3 base pairs down here. How this happens is that it needs to become activated within the enzyme amino acyl synthetase. So this enzyme, there's my transfer RNA, he's coming in. My amino acid is going to come in. They fit together perfectly. This is where energy is now going to play a factor. So ATP is going to come in and it's going to bond my tRNA and my amino acid together. The enzyme is helping to speed up this process. Once they become bonded together, my ATP, I broke off 2 phosphates, becomes adenosine monophosphate, low energy molecule. My enzyme breaks off. I now have an activated transfer RNA with its amino acid there. The amino acid is specific to the anticodon on the end or the bottom of my transfer RNA. So now what has to happen is that this transfer RNA is going to make its way to the ribosome, and it wants to find its codon. This is my anticodon. So my anticodon is GGC, and the codon that it's going to match up with is CCG. And they're going to match together perfectly. And when that does that, it's going to come in, it's going to attach to the A site of my ribosome. The next one's going to come in, and it's going to kind of push it to the P site. And we're going to start building, and my amino acids are going to start bonding together. You guys will see this in the diagram here in a moment. So when that messenger RNA enters into my ribosome, this is called the initiation. So what's happening during initiation is that the ribosome is reading the start codon. And like I said, there could be hundreds of base pairs, but until that ribosome reads AUG, nothing happens. Once it reads AUG, it stops, the messenger RNA stops in that A site, and I have started initiation. I've read the start codon, and I am ready to go through and make my protein. Eventually, more and more of these transfer RNAs and my amino acids are going to keep coming in. Goes into the A site, everything shifts down, they go to the P site. My amino acids are going to keep bonding together, and I'm going to get a string of these amino acids. A string of amino acids eventually is going to be called a peptide or a polypeptide, and they're joined by that peptide bond. As I am growing my amino acid chain and I keep adding and adding and adding more of them, that step is called elongation. So start codon is initiation, that's my first step. My second step is elongation. Eventually what's going to happen is that I'm going to reach what is known as termination. So I'm going to go back. Here's my little messenger RNA. Let me see if I can blow this up for you guys. Here's messenger RNA. Here's my 2 subunits of my ribosome. And as I go through, what you guys are going to see on this particular one right here, here's my messenger RNA. Here's my ribosome, big subunit, small subunit. I have an A site. I have a P site. This E site here, you won't see it on the ribosome that I'm having. That's just the exit site. That means where transfer RNA is breaking off. So like I said, you'll see ... here on messenger RNA. I could have had thousands of base pairs before then. Until that ribosome reads AUG, nothing happens. Once he reads AUG, he's actually starting in the A site. He shifts over a little bit. He starts calling out, hey, AUG, 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 bring in the correct transfer RNA. Transfer RNA anticodon is going to match up with the codon. So a messenger RNA, A goes with U goes with A, G goes with C. Codon is complementary to my anticodon. On the other end of this transfer RNA, I have my amino acid. Here it happens to be methionine. The ribosome reads my next codon, UGG, starts calling it out, wants the appropriate anticodon to come in. So in comes U goes with A, G goes with C, G goes with C. That one comes in here. My 2 amino acids are now going to bond. The bond is going to break between transfer RNA and my amino acids, and my 2 amino acids will bond together with that peptide bond. So both sites are going to be full, my P site and my A site. That ribosome wants to read my next codon, UUC. So what happens? What's in the P site slides down. It's actually an exiting site. So this transfer RNA that we've been talking about here is actually going to break off. So transfer RNA breaks off, he leaves behind his amino acid, he goes out, he gets another one. What was in the A site goes to the P site. Now the A site is open. In comes another transfer RNA. U goes with A, U goes with A, C goes with G. I attach another peptide bond, I join another amino acid. That is called elongation. Eventually, as I keep going through and adding more and more of these amino acids with tRNA, I'm going to reach something called a stop codon. And that leads us to our third step, which is called termination. So there are 3 codes in messenger RNA because remember, he got his code from DNA that said, hey, this is the end of it, this is where it stops. UAA, UAG, and UGA are your 3 stop codons. They do not code for an amino acid. There is no transfer RNA. So UGA, which is a stop codon, there is no transfer RNA that says A, C, U. There's nothing that says that. There's no amino acid that goes there. So it will actually stop that particular chain. It says, okay, this is all I need to make that enzyme. This is all I need to make that protein. And at that point, everything's going to kind of break down and my protein will be packaged for use. So, once again, when that ribosome reaches a termination codon, and like I said, there are 3 of them. They're listed right here for you guys. The polypeptide, which is my protein, is cleaved or broken off from the last transfer RNA. Transfer RNA, they're empty, taxi cabs. They have no passengers, no amino acids attached to them anymore. They go back out into the cytoplasm, they pick up the same one that they had before. My ribosome is actually going to start to break down. Small subunit, big subunit will actually break down, and my messenger RNA can kind of be broken down to have those parts recycled. Or if you need more of that enzyme, they're going to go find another ribosome that's activated and go through and make more of it. So at that particular point, my protein is ready to be packaged. And if it's packaged, it can either travel through the endoplasmic reticulum. We'll talk more about this tomorrow. Will go through the endoplasmic reticulum, make its way to what is known as the Golgi body, and then either be shipped out of the cell through active transport or it can be utilized within the cell, depending upon what it is. So maybe it's a little protein that's on the cell membrane that we need. Maybe it's a protein that takes place with digestive enzymes that have to be released in the stomach, or salivary amylase that gets released in your mouth. It's getting pushed out through little vesicles out of your cells and making its way to other places. That takes energy to do. So if we think about this entire process, I'm going to go more towards picture view. Let's go here. So if it looks like there's a lot, there kind of is. Let's split this up. So this first part right here just shows the difference between your codons and your anticodons. So this whole page that you're going to see here with your coloring goes through the process of translation. I will do tabletop protein synthesis again, just like I did with replication. So I know some of your comments from transcription, hey, if I saw an animation, you're absolutely right. Unfortunately, Flash is no longer covered on our computers. Flash kind of got taken off for all your animation, so it's more video that you guys are having. And I wanted to get through the second part, let that sink in, and then we'll see more animation videos that will help that, including my tabletop version of it. 2 steps of protein synthesis, transcription and translation. Transcription, making messenger RNA in the nucleus, we're making it from DNA. That's why it has to be in the nucleus. Translation, we are now reading the messenger RNA at the ribosome in the cytoplasm. So translation is involving all 3 types of RNA. So here we go. My difference is in messenger RNA, every 3 base pairs is called a codon. In transfer RNA, every 3 base pairs is called an anticodon. I have them divided into 3 steps. So here's the first picture is just going to show kind of initiation and start codon. Then we'll go through elongation. Then we'll go through termination. So when we have initiation, here's my messenger RNA. Here's my small subunit of my ribosome. So messenger RNA is going to find a small subunit and it's going to become activated. What that means is that now that my large subunit, my small subunit, they kind of click into place with our RNA. Notice that it's my 5' end of messenger RNA that entered into the ribosome first. That's going to get read first. And it goes from the A site to the P site, the peptide site. At that point, the ribosome starts to yell out, hey, I have a codon, AUG, AUG, that's my start codon, come on in, fill that spot. And what comes in is my transfer RNA, this blue structure here, with my amino acid on the reverse side of it. A goes with U goes with A, G goes with C. Codon, anticodon, complementary, lock into place. The ribosome now reads what's in the A site. I can only fill 2 spots on that ribosome at a time. So within that A site, now the ribosome starts yelling out, hey, GCU, where's my complementary anticodon? G goes with C goes with G, U goes with A. And that specific transfer RNA is going to come in and bring his specific amino acid as well. So remember, these amino acids and transfer RNAs became activated out in the cytoplasm as well using the enzyme amino acyl synthetase. That means that they became activated. They hooked together. Transfer RNA, amino acid, these are just free floating. This comes from the food that you eat, your amino acids. There are 20 different amino acids floating around. Your transfer RNAs are now picking them up. They become activated at that enzyme amino acyl synthetase with the help of ATP, because I need energy to actually physically bond them together. I'm creating a bond which stores energy. That energy is coming from ATP. Now this is what you are seeing right here, coming down to that transfer or coming down to that messenger RNA. That's elongation. As we move down, we go down to, that was initiation. As we move down, we go to elongation. It just means we're growing the polypeptide chain. We're getting bigger, we're getting longer. Here was my first one right here. That was in my P site. We said, okay, well now the ribosome's reading my next codon, GCU. Here comes my complementary anticodon. G goes with C goes with G, U goes with A. He's going to come in and lock into place. What's going to happen now is my 2 amino acids are going to bond together. They are creating a peptide bond between 2 amino acids. Everything shifts down. What was in the P site gets kicked off. So what was here now breaks off. You can see he breaks off right down here and leaves behind my amino acid. What was in the A site shifts over to the P site. Now I have an open activation site. In it comes UUU, here comes AAA, he's got his own amino acid. They will bond together. Elongation, I'm growing the amount of amino acids that I have there. Eventually, let's say we have a really long chain, that ribosome is going to read one of 3 stop codons. Here it happens to be UGA. It could also be UAA, UAG. Does not code for a transfer RNA. There is no anticodon that matches with it. There is no amino acid that's going to go with it. So at that point, another enzyme is going to come in and say, okay, this is in, let's end it, let's stop. Termination. So initiation, we read the start codon. Elongation, we're growing that whole process. Termination, we're ending it. We said earlier, our smallest protein, though, is about 50 amino acids in length. This is just a really short version of it for you guys. Everything then begins to break down. So my ribosome can actually break down. It can be recycled. My messenger RNA, the same thing happens. If I need more of the same enzyme that I've just created, it's going to go find another ribosome to activate. Transfer RNAs, they're just going back and picking up more amino acids that we have there. Now I'm left with this polypeptide, this protein, joined together by peptide bonds. It can either be utilized in the cell for, let's say I need some helicase for my DNA replication, or I need some ligase to glue together my Okazaki fragments. Or it can be used outside the cell. I need little protein receptors on my cell membrane. I need to make amylase so I can get released in my mouth. So the cell's going to figure out, am I packaging it here, or am I packaging it to get shipped out? And that's what the endoplasmic reticulum is going to do, and that's what your Golgi body is going to do. So the endoplasmic reticulum, your rough endoplasmic reticulum that has the ribosomes on it, it's rough because it has the ribosomes. It's kind of like roadways throughout the cell. So it's transporting that protein to the next step. And the next step is going to be the Golgi body or Golgi apparatus. That Golgi body or Golgi apparatus, I say, is kind of like the big UPS warehouse. You guys order something online, that company ships it, they ship it to UPS, or they ship it to Amazon, or they ship it someplace else, and then they kind of repackage it, redo it together, and then ship it out to where it needs to go. That's what the Golgi body's doing. He's getting your protein from the endoplasmic reticulum. He's going, hey, am I using it in the cell or am I using it outside the cell? Oh, I'm using it here. Okay, let me do this to it, package it in this kind of little formation. Maybe it's going to be a lysosome to help break down cell parts because it contains a digestive enzyme. Maybe I got to put it in a little vesicle so I can ship it out of the cell and be utilized outside the cell. So I'll give you guys a little more of a diagram on that tomorrow as we reiterate everything that we have here. So 3 steps of translation: initiation, elongation, and termination. So the page that I showed you guys the other day, the little fact sheet, the second page on it is actually your translation. It does go through and label, so you can see here your codons, your anticodons. So it says the ribosome binds with the start codon, messenger RNA. So you can see, and I like this one because there are base pairs before it, and it doesn't do anything until it reads that AUG start codon in that A site. It's going to shift it to the P site, and that's what's going to allow it to call in the transfer RNAs. So in comes my little transfer RNA. He's got his little anti-codon there. It's going to match up with my codon, complementary base pairs. No thymine anywhere out in the cytoplasm here. So we have gotten initiation. We now are going through elongation. So as my transfer RNAs come in, it's kicking off the previous one because they can only hold 2 transfer RNAs at a time at that ribosome. One in the P site, the peptide site, because that's where that bond is being created. One at the activation site, which is where that transfer RNA is first entering onto that messenger RNA strand. This is elongation because as that transfer RNA falls off the ribosome, he's leaving behind his amino acid. It's joined by a peptide bond to the next one, joined by a peptide bond to the next one. Remember, 2 amino acids is called a dipeptide, a whole bunch is called a polypeptide. So here's my elongation because we're growing that polypeptide chain. Eventually, it's going to reach a stop codon. So in this case, we can say UAG, could be UGA. There's 3 of them there. And at that point, the ribosome releases, he kind of lets go of the messenger RNA. There is no transfer RNA that's going to go through and say, okay, I need to hook up here. There's no codon to anticodon pairing for that stop codon. At that point, the amino acid chain, we showed you 3 amino acids together. We said the smallest one's going to be 50. It can start to fold. It's going to become a protein, and that's where it's going to get packaged and shipped out to where it needs to go. Okay, so I know this is a lot of information. I know there's a lot going on with it. Tomorrow, when we see each other for notes, I'll go through the tabletop version of it. I'll go through a little animation video of it because I know just seeing here on paper, seeing it here, if you guys are in class, we absolutely will go through the molecules and models. They'll be out on the table. So if you're like, oh, today's an A day and I don't have you till B day, they'll still be there. So if you guys want to go through it with me and explain stuff, if it helps that way, please do so. Okay, so we'll let this sink in. We'll do it again and again, and we'll make sure that everything is solidified with